Thanks so much for the invitation to present my research lines here in the Matos workshop. So I'm Luis Díaz. I'm part of the Department of Pharmacology, Pharmaceutical Technology here at the University of Santiago, and also a member of Matos. And my work, the work I'm going to present today, it's about the additive and subtractive biofabrication for tissue regeneration. So my field is tissue engineering, and as you may know, this is what we call the triad of tissue engineering, which is the combination of scaffolds, cells, and biochemical factors to provide an ideal environment for tissue regeneration. So to talk about the capabilities and the abilities of that I acquired with the time, I'm gonna divide my work into the three main areas. First, the biofabrication, also biological studies, and then structure characterization. So to talk about biofabrication, first I want to mention what are the capabilities that we have here at Matos. And this is the 3D printer called 3D Bioplotter from EnvisionTech, and it's probably one of the most advanced 3D printers that are available for biofabrication. And we have it here inside this building, and it's inside also a flow cabinet, so we can print not only cells, fuse deposition modeling, etc. But this printer is very interesting because it has 5 heads, so we can incorporate up to 5 different compositions into our scaffolds, into our constructs. So that means we can combine cells printed inside bioinks like hydrogels with fuse deposition modeling, with backbone polymers that we can print and deposit like PCL or PLA, etc. And it's very interesting because of that. It's very easy to use, and it's available for all of us to use. So the goal of my research, or one of my lines, is the regeneration of complex tissues. Most tissues are very complex, heterogeneous, and it contains gradients. One of these tissues, the one I'm mainly focusing, is the osteochondral unit, which is composed of the cartilage, articular cartilage, and the subchondral bone. So within a few millimeters, 2-3 millimeters, we have very different extracellular matrix arrangements with different collagen fiber distribution, pore sizes. In the surface area, we have a very tight extracellular matrix composed mainly of collagen, with pores around like 100 to 100 microns. And then as we go down into the bone, those pores, they open, the arrangement of the collagen fibers is different, and in the end, we have the subchondral layers, which are calcified, containing hydroxyapatite, which is the main calcium salt of the bone, and then to the subchondral bone, which has pores up to like 1 millimeter, and it's mainly 70% calcium content. So in order to replicate this structure, we can use that printer, and that's what I've been doing during my postdoc, to replicate both the composition, specifically the calcium content, and the pore size. So to do that, we can do vertical gradients, which is basically achieved by every layer that we print. We combine different compositions. If we do a gradient from the subchondral bone, we have a high content of calcium, and then as we go up, as we grow the scaffold, we can decrease the fiber space, the pore sizes, and also decrease the calcium content. So we can modify that on every layer. The size of the layer depends on the diameter of the scaffold of the needle. So we can just modify or do these gradients with, for instance, 200 microns if we use a needle of that diameter. But we also can do horizontal gradients. So we can basically create a scaffold that on every layer contains, for instance, in this case, 3 different compositions and 3 different pore sizes. The way to do that is to combine or to bind segments printed with different compositions and also with different needle diameters. So the fiber thickness is different, and we combine the 3 of them within the same layer and we grow up the scaffold, what we have is 3 different regions that are distinct in composition and porosity, and we can also combine these horizontal gradients with the vertical gradients. So in the end, we have control of all the axes of our construct. So if we do, for instance, in this case, 3 different scaffolds controls with only one composition, in this case, we have a gradient of calcium content from 30% and small pores to 15% hydroxyapatite and medium pores and large pores with only PCl. And in the end, we combine them in the same scaffold. We see that there are no difference of in terms of like porosity, fiber thickness of the individual scaffolds, control scaffolds, and those regions of the gradient scaffolds prepared with those same setups. And we can also use that to decorate our scaffolds, our regions of the scaffolds with specific peptides, for instance, in this case, using click chemistry, that cells, when they attach in those regions of those scaffolds, can sense those peptides and differentiate into specific cell lines. For instance, if we use N-cadherin, those cells will turn into chondrocytes, and but only in those those regions of the scaffold that are decorated with that. And in the other regions, for instance, we can incorporate BMP2, for instance, a bone morphogenetic protein that drives the differentiation of stem cells into bone, and we can get those different cell lines or those differentiated cells without combining different culture mediums or anything, just by sensing those surfaces. But all of these that I just talked about are only useful for, or as I mentioned, only for like PCl, which is fused deposition modeling. We have to print temperatures above 100 degrees, so it's impossible to incorporate cells into that. Cells will die. The way to do that, to incorporate cells also in this kind of scaffolds, the traditional method is using a co-printing. So we can print a polymer backbone, for instance, of PCl, and then in parallel, we can print fibers with our bioinks that contain the cells. The problem is that if we have to incorporate those bioinks into those scaffolds, we still need a high viscous bioink that can decrease cell viability just because of the shear stress at the top of the needle. And also, we decrease the porosity. We are filling those spaces with these bioinks. So we came up with the idea of using those backbone fibers to serve as beds, as molds, to print our inks in there. So the way to do that is we print every layer of, in this case, for instance, PCl, and then we engrave those fibers with another cartridge using an empty needle, heated. So we can just do this engraving, but by remelting the surface of the fiber, and we create that bed that we can use afterwards with another head. Remember that we have like 5 different heads, 5 different compositions, and fill those beds with our bioink. We don't decrease the porosity, and we don't need high viscosity in the bioink because we have that area contained, that volume is contained. So we can take advantage of that to print, for instance, cells. We can fill those gaps. So this is how those grooves look like. We can print cells, or we can print also other materials. So we can just fill those gaps, those grooves, with any material that we have in our mind. The next step, and this is one collaboration that started up here in the Matos with Professor Maite Flores, is instead of using those needles to create the grooves, using laser, femtosecond laser, we can use that to create with high precision those grooves. But we cannot only create grooves like these ones, like these ones, that we can do, or until now, we were able to do it with 20 micron precision, 20 micron diameter, but also we can create another patterns, like honeycomb or like a spotlight. Fill every layer with this kind of patterns that I hope next year we can show you results that are really interesting, how cells behave with these different patterns. And the second goal or second capability that I want to talk about is the biological study. So apart from like doing in vitro studies, we do cytocompatibility, cell proliferation, differentiation, like PCRs, etc. I want to talk about the CAM model. So in order to assess the suitability of a scaffold before going to in vivo, we need to check first how it integrates with the tissue and also how it vascularizes. So how new vessels grow inside the scaffolds, because we need those goals to improve or to success in our goal of tissue regeneration. So one way to do that, is the chorianthogenic membrane aside. So it's an in vivo embryonic system, so it's pretty replicable. It also complies with the 3R approach, so reducing the number of animals, etc. It's very accessible and it's very affordable. So the idea is that we can use the chorianthogenic membrane, which is the upper membrane of the egg, which is just behind or beneath the eggshell, and it's very interesting because it's very vascularized, and we can use this kind of studies until day 16 of the embryo development until it becomes sentient. So we don't need to apply for any committee or approval for the ethics committee. The idea is that we fertilize eggs, we incubate them. After 3 days, we open the window to expose the CAM, and on day 7, we place our scaffolds. Can be scaffolds, can be gels, can be any kind of material in contact directly with the CAM, and then we return into incubation. And then, depends on the day, we normally do it until day 14. We allow 5 or 7 days of incubation. We evaluate what happens with the scaffolds, with our material, in contact with that membrane. And first, what we do is a visual inspection to evaluate if there is any cytotoxic events in the tissue surrounding the scaffolds or the material. Then we do integration score to evaluate how that membrane, the extracellular matrix, grows into the scaffold or it's in contact with it. And then if we do angiogenesis studies, we also count the number of vessels that grow radially around the scaffold in order to evaluate these angiogenic effects. And then the third one is the structural characterization. Here at the University of Santiago, we have, as Matus members, we have access to 2 micro-CT equipment. We have a materials equipment which has a resolution of 500 nanometers, and we have an in vivo micro-CT which has a resolution of 50 microns. So for the acquisition of those images, that comes with the USC, but for the rendering and the quantification, something that I also been trained during my postdoc. And what we can do is evaluate different parameters of those samples that we evaluate. Can be polymers, can be animals, can be tissues, can be glass, materials, different kinds. And we can evaluate the structure in terms of individual analysis, how its distribution, the porosity, the interconnectivity, and also that distribution of the composition. Here we have 3 examples. For instance, in the field of engineering, these are scaffolds printed with this horizontal gradient, so we can evaluate that different porosities, etc. Also, like in the pharmaceutics field, we can also do, like for instance, with a tablet, we can evaluate the drug distribution, pore size, how thick is our coating. Or also, I've been doing also studies with brains, for instance, with mouse brains. You can also evaluate what are the differences or any structural differences in the mice depending on which drug treatment we are using. So one last thing, we can also do that. Remember the CAM studies. We can also use the micro-CT to evaluate First, the tissue integration, so we can calculate what is the volume of tissue that grows inside the scaffold after we incubate it with the egg. And also we can evaluate devascularization. We can stain the blood vessels, and we can do a 3D reconstruction, and we can calculate how many and what is the size of those vessels that grow inside the scaffold. So in the end, the 3D printing is a very powerful technique to fabricate the scaffolds and create gradings in all directions. The CAM is a cost-effective technique for the screening of our materials before going to in vivo. And using micro-CT, it's very also useful because it's non-destructive, and we can enable, or it enables the precise evaluation of 3D structures in different ways, and also tissue, ingrowth, and vascularization. So thanks so much for your attention, and if you have any question.
Una curiosidad.
In English or Spanish? Okay. Thank you very much for your nice presentation. I have a naive question related with the films or capsules. What is the limit if you want to open a hole, the capsule like vessels? What is the minimum diameter of the hole of the open?
You mean in the egg?
Yeah, with the
Depends on the size of the material you want to incorporate.
biopolymer, alginate, or no?
If you want to just, if it's like injectable, syringeable, you can just open a very tiny window and then deposit your material in there. And then after 7 days, you have to open it all just to expose and check the material. But it depends. As long as your material can goes inside that hole, it's enough.
In your case, it's around 200 microns, 500 the diameter of the...
Of our scaffold?
Yes.
No, they are like 8 millimeters.
8 millimeters.
Between 8 and 10.
Yeah. Okay, thank you very much.